Now that you know that liquid biopsies are cool, you shouldn't give up on pathology. Actually, tissue pathology is still important. And our next speaker, Dr. Miguel Rivera, is an assistant professor of pathology at Mass General. And one of the challenges is that the number of markers is rapidly outpacing the number of good antibodies to analyze tissues. So Miguel will talk about RNA methods of staining for tissues. Miguel.
Hi, thanks for the introduction, Daniel, and thanks for inviting me to talk about our work. I am a pathologist, an anatomic pathologist. I also run a basic research lab at Mass General. And the story I'm going to tell you about today is about a tool that we've been using in the basic research lab, which we surprisingly found we could translate to the clinic relatively fast and efficiently. So this is RNA diagnostics, or RNA-ish. I think of an example of something that has been readily translated. This is probably one of the best examples in pathology. So I'll show you some what our experience has been. As a pathologist, we're trained to look at tissues and interrogate them with a number of techniques. And the most common one is immunochemistry for looking at proteins. But essentially, immunochemistry is the king of all these techniques because it works very well in paraffin. And we have, at different times in our history, tried to move away from paraffin and try to collect frozen tissues or collect different types of materials. But truly, paraffin is the most efficient way of storing and collecting patient samples. Different hospitals have tried to move into a full collection of frozen tissues and have had practical problems, from having difficulties establishing a large number of refrigerators where you can store samples at -80, for example, to practical issues where in a busy pathology service, somebody would have to put some tissue in formalin for paraffin, as we always do, but then also think about keeping some additional tissue for other techniques. So when thinking about biomarkers, really, if you have something that works in paraffin, you're way ahead. So that's why immunochemistry is so powerful, because we can actually work with proteins that are fixed, but that we can retrieve using a number of antigen retrieval methods and actually interrogate in the tissue section, looking at the cells the way we like to do in pathology. However, immunochemistry has a number of downsides, and the most important one is that it relies on antibodies. Antibodies, of course, are great, and when you have a great antibody, you can really detect a protein very efficiently. But developing antibodies for a large number of antigens is actually a really cumbersome process, and there's a number of companies that do a good job, but they get stuck in several classes of proteins that are difficult. One area that I've worked on in the research lab, for example, is Wnt, and we don't have any good antibodies for Wnt. Wnt are secreted proteins, they're really small, they're all similar to each other. There's really no good antibodies to do this in tissues. Another type of biomarker that, of course, we use a lot is DNA, and this is in the molecular diagnostics lab where I do most of my clinical work. We use DNA a lot. But DNA really limits you to looking at mutations, and it's very powerful, but you cannot look at the functional status of the cell in terms of what genes it's expressing and what is it doing. Is it dividing and whatnot? There's some great markers that we can use in the basic research lab, but that we cannot use in the clinic very well. So what about RNA? RNA is a molecule that we are used to interrogating in basic research in cell lines and in frozen tissues. And in fact, in the area that I work on, which is the connection between developmental biology and cancer, we use RNA in situ to detect genes, particularly when we don't have a good antibody. And this is a technique in the lab that only the most brave postdocs will take on, because it's a really unreliable technique. So we had a number of markers, and this is how the story began, that we were interested in, particularly new genes that we had cloned, that we couldn't efficiently detect in even in basic science, perfectly prepared tissues. And what we realized is that the problem was that for RNA in situ to work using normal techniques, you have to make RNA probes. And RNA probes are incredibly unstable. So what we did is we partnered with Affymetrix, and they had this technology, this was several years ago. They had this technology that is based on branched DNA amplification. And it has really two aspects that I think are really important for the detection of RNA in tissues. The first one is that it's based on oligonucleotides that attach to the RNA. And oligonucleotides can be synthesized very readily. You don't have to clone your probes anymore. And they are DNA, so they're very stable. So we are, with this one step, we already are way ahead of where we were before, which was easily degradable RNA probes. Now we have very stable oligonucleotide probes. And the second part is that it uses an amplification based on branch DNA, which greatly amplifies the signal in a linear way, so we maintain the relationship with the expression levels and the signals that we detect. So we quickly started testing this in the research lab, and the results were great. We got really nice stains like this one. So this is tumor tissue where we have the epithelial tumor cells nicely expressing RNA for an epithelial marker. And this was very nice because we have what we don't often see in immunochemistry, a very good signal-to-noise ratio where the positive cells are clearly positive and the negative cells are clearly negative. Our surprise was that when we went to expand this study, we collected a number of tissues from the Mass General archives, paraffin-embedded blocks, and interrogated blocks that were 10 years old, 15 years old, and we got exactly the same result. So this technology let us not only improve our performance in the basic research lab, but unexpectedly in the clinical lab. And the reason for that turns out that paraffin-embedded tissues are actually pretty well fixed for RNA. They're not well fixed for RNA and long RNA molecules, but there's some amount of degradation that happens. But because you're detecting the signal using short oligonucleotides, it doesn't matter that their RNA is a little bit degraded. There's still enough for you to see the signals. So this is actually a somewhat unexpected situation that actually makes clinical tissues relatively easy to interrogate if you have a powerful RNA detection technology. And now there's a number of different approaches to do this. And we've continued using the same branch DNA technology that we started with. We're very excited about its performance. And so because of this result, we went to some of our colleagues in pathology and the cancer center, and they were very eager to try some of the things that they hadn't been able to do before. So there's some very basic markers that actually are surprisingly useful that people know we would like to interrogate, but we haven't been able to. So the first one was, this is actually a GI pathologist that we work with, who is now a member of our team, Vikram Deshpande, told us that in gastrointestinal pathology, we've always known that the best marker for liver origin of cancers is albumin. But the problem with albumin is that albumin is a secreted protein. In a secreted protein, you cannot detect very well by immunochemistry because it's usually just all over the place. You see brown stains everywhere, and it just doesn't stay in the cells. If you have RNA detection for albumin, you have a completely different picture. This is an example of a hepatocellular carcinoma, which has very strong stain for albumin. And in this particular case, the other markers that were attempted for liver origin were negative, whereas the RNA was clearly positive, very strong, clearly positive. So this really facilitates cancers of unknown origin, for example, where you want to know if you have liver origin of the cells. So this array has been published, and it's actually in clinical use right now based on these studies. The second category that was surprising and interesting was to detect viruses that are related to cancer. So HPV, as many of you know, is related to squamous cell carcinomas, and there's a lot of research activity in this area. And the test that we have used is to detect DNA in these tissues. And that's a fairly useful test, but RNA turns out to be much more abundant when you have expression of, when you have HPV-infected cancer. So it's a much more easily detectable molecule if you can if you have a good technique for it. So we found that we have a lot more sensitivity detecting HPV RNA in clinical samples, and it has worked very well for us. And this is, again, entering clinical practice right now. And finally, this is a category of molecules that you can only interrogate with in situ RNA technology because it's an RNA. Line 1 is, for the most part, an RNA. Now, there are line 1 contains some coding regions, but only a minority of line 1 molecules actually create proteins. But the genome has a large number of loci that are producing RNA constantly. And so we can detect this very well in tissues, in clinical samples, and we have noted, and other, there's a number of publications now raising the interest in line 1 because this has been related to immune responses to cancer and also to the levels of acetylation found in the genome. So it's an interesting biomarker that, again, RNA has some very strong advantages for. So finally, what we have done, and this is where we are now, is to try to set up an RNA biomarker discovery lab at Mass General that's set up for implementing on a large scale a number of translational projects that are going to use this technology. And as I mentioned before, there's a number of advantages in terms of the types of biomarkers that we can interrogate. And particularly, we have an automated platform where we can do this relatively quickly. Thanks.